Hi, my name is Kyle McCulloch, and I am a research assistant professor in the Department of Ecology, Evolution, and Behavior. And I identify as a cis gay man, and my pronouns are he, him, his. And I study the evolution of visual systems in animals. And so here I'm just showing some of the diversity and the variety of different types of visual systems and eyes that we see in the animal kingdom. And what I'm drawn to by this is just the spectacular diversity we see in animals, as well as the beauty and complexity of these structures. So currently, I don't study any of these. I study this sea anemone called Nematostella vectensis to understand the evolution of visual systems. And so why study eyes in the first place, and why study an animal without eyes? And so eyes are just a really powerful model to study the link between genes and traits. And it's possibly one of the best we have for this. And so animal diversity allows us to compare this link across many different contexts in these related systems. And there are two really important aspects shared by all animals. And we can link from gene all the way up to trait. So one of these is developmental genes. So these are genes that turn on cells in development and lead to very different eyes. So the same genes lead to either the human eye or dragonfly eye, and this is shared by many different animals. So these are similar genes that act in different contexts to produce very different traits. The other gene that is related in many animals is the actual gene that does the light sensing. So this gene is found in the retina of all animal eyes, and they're known as opsins. So this is a family of proteins that detect light, and it's what all animals use to see, which is a very important sense, such as for looking for mates. And so why study the anemone? Well, this is because complexity can take many different forms. So what we think of as complex in the human eye, there's a complex structure, whereas a sea anemone has no eyes. The human eye is made up of multiple different tissue types. We know that there are many, many photoreceptor cells in the human retina. We don't know much about the photoreceptor cells in the sea anemone, but one thing that the sea anemone has on the human eye is in our eye, we only have 4 of these opsin genes, and the anemone has 30. And so it's very interesting to look at how these have expanded and the photoreceptor cell type evolution of these cell types, and to compare that to other visual systems such as humans and other animals. And how do I do something like this? Well, I'm looking currently at how cell types evolve and diversify. And so we look at cell development, shape, tissue type. We look at gene expression and regulation, photoreceptor cell physiology, photoreceptor function, and behavior. And the way I do this is I will knock out genes. I do animal behavior experiments, a lot of gene and genomic sequencing. We use fluorescent transgenic reporters, and we look at and measure individual cell physiology. And what is the importance of this? Why study these weird visual systems? Well, it's helpful to understand the rules of how genes interact and to build a trait across many different contexts. It's also useful to understand the avenues available to evolution and the constraints, so how evolution proceeds and how it can or cannot proceed. And all of this together actually broadly helps us understand our own genetics and disease, so many eye diseases. And so I hope that you enjoyed the talk. I thank you for listening, and enjoy the other talks. Thanks.